Morning, all. So this chapter is chapter 3, which is tools of the lab or tools of microbiology. And we're going to go over a lot of the stuff that you would typically be doing in a lab if, within a microbiology lab. Pretty much cultivating these microorganisms, various tests to identify these organisms, and a lot of these concepts will actually cover with the lab's exercises that you do have on the online course. Okay. So when we look at microbiology, there are 5 basic techniques that we use to manipulate, grow, examine, and characterize the microorganisms in the lab. This will include inoculation, which is introducing the organism to, say, a culture medium. Incubation is creating the appropriate conditions for the organism to grow, whether it's a certain temperature, pH, atmospheric gas concentration. Those are all factors that need to be taken into consideration when you're looking at incubation. Isolation, this is very important for clinical labs. A lot of times when you take a sample from a patient, for example, that particular sample is not going to be homogeneous, meaning just one thing. It's probably going to be heterogeneous, which means it's going to have a lot of different organisms present. And you're going to have to sort of isolate out the one that you want. Inspection and identification, these are also very important for clinical labs. In realistically, a lot of clinicians want to know what it is, because determining what it is will determine the mechanism in which you're going to go about treating, say, an infection. Okay, so for the first one, inoculation, this is producing a culture. A culture is when you cultivate microorganisms. Cultivation means to grow. Any growth that appears in or on the medium after incubation is considered a culture. And hopefully it's the culture that you want and not a contaminant. The medium, this is actually a nutrient-containing environment. It could be a petri dish, for example, or it could be a liquid medium. And this nutrient-containing environment is where the microbes actually can multiply, okay, and grow. You're essentially creating an environment for these organisms to grow. Inoculation is the process of introducing the microbes into the media for culture. And generally speaking, when you inoculate a media, you want to do it aseptically. Aseptically means without contamination. And we usually say that this is a sterile transfer, okay? Sterile transfer, your textbook actually defines it free of microbes. I'm not 100% sold on this particular statement right here. And I want to point out something. It should be free of living microbes. In reality, you will sometimes have media that is actually made up of dead microorganisms. So they're there, they're just not living. And then a requirement for any instrument used for sampling or inoculation should be sterile. Okay, meaning it should not be something that you just picked off of a lab bench. If it is, most people will sterilize that by passing it through a flame. Okay. We also have a lot of sterile plastics that can be used. The disadvantage to using sterile plastics is that they are one-time use. So you use the sterile loop and then you would toss it. Generally speaking, most people will use metal loops. Metal loops are things that can be reused over and over again. And what you end up doing is you will end up sterilizing the metal loop by essentially holding it in a flame to make sure that there's no living organisms or microbes present there. Okay, incubation. Incubation is, again, setting up the appropriate conditions for these organisms to grow. One of the biggest factors is temperature. And the typical range for growth for most microorganisms is going to be somewhere in the realm of 20 to 45 degrees Celsius. You go below 20 degrees Celsius, growth will probably slow down significantly. You go above 45 degrees Celsius, you're probably going to start to kill cells. And the reason why you would end up killing cells at a temperature above 45 degrees Celsius is because that is going to be the temperature at which you start to denature proteins within inside the cell. And when you denature those proteins, unfortunately, you're going to lose a lot of critical pathways and the cell will just die. A lot of incubators will also be able to control atmospheric gases, such as oxygen and carbon dioxide. And you might need to control that or set certain percentages in which those gases comprise the air that the organism is growing up in. Microbes must be provided with all of their required nutrients in an artificial medium. Some microbes only require a few simple inorganic compounds to grow. Others can be very complicated and require complex lists of specific inorganic and organic compounds. Certain microbes are not able to grow on artificial laboratory medium and might actually require cell cultures or host animals. This is particularly true for viruses. You're not going to be able to take a virus and culture it on a petri dish for the main reason that a virus is non-living, and viruses must have host cells in order to replicate. So in order to replicate viruses, which means to make new virus, you would need a host cell that becomes infected with the virus so that it's able to replicate that virus. Okay, so I mentioned a little bit about sterile technique. Sterile technique is also referred to as aseptic technique. And this is absolutely necessary when inoculating media. Media and inoculating tools must be sterile. Measures must be taken to prevent introduction of non-sterile materials, such as room air and fingers, into the media. Your surfaces do contain microorganisms, and if you were to touch the surface of the media, you would contaminate it. Unfortunately, there can also be microbes and spores that travel through the air. And as a result, sometimes just by opening a plate, a spore or an organism may land on that plate, and that can cause contamination. It is very important that when you are culturing, that you do not leave the plates just open, that you only open the plates when you are actually actively, say, inoculating it, and then you immediately close it. You just don't want to leave a plate open on a lab bench because it will open up the door for contamination. Now, there are different types of media. We have defined or synthetic media. Defined or synthetic media is media in which we know exactly every type of chemical that is present. It can contain pure organic or inorganic compounds. It will have molecular content that is defined by means of an exact formula. And this is something that's used in research a lot because they want to really control every aspect of the experiment. Complex media, on the other hand, this is one in which at least one of the components is not chemically defined. Okay. Now, we typically see most media being complex media, especially if this is just plain old culture media to grow up a microorganism. And what we mean by not defined is the recipe for, say, a complex media might call for 10 grams of protein in a liter of, water. You're putting in 10 grams of protein, but you're not really defining what that protein is composed up of. After reviewing chapter 2, you should be familiar, and hopefully from a prior biology course, that proteins are made up of amino acids. There are typically 20 amino acids. If you were just putting in 10 grams of protein, you don't know exactly the content of each amino acid that's present there. Okay, you just know that you have 10 grams of protein, which is a very good nutrient-rich environment. However, if you were making a defined media, you could not say 10 grams of protein. You would actually have to define out each individual amino acid and how much of each amino acid is actually present. So the difference between the two types of media is one is very specific and every condition is accounted for, where the other is not. And obviously you can imagine cost-wise, defined media is going to be a lot more expensive. So we typically use complex media for just generally growing up microorganisms. It's usually very nutrient rich. It has a lot of microorganisms present. And I'm sorry, it doesn't have a lot of microorganisms present. It has a lot of nutrients present, which will allow a lot of different microorganisms to grow up. Okay. So those are the two main types of media, and there are many subdivisions within those two types of media. For example, we have general purpose media. This would be a complex media. This would grow a broad spectrum of microbes. It's complex media that contains a mixture of ingredients that will support a wide variety of microbial life. Enriched media is very similar to general purpose media. And what this is that it actually might need particular additives to the media. For example, blood, serum, hemoglobin, certain vitamins might actually need to be added to the media so that the organism can grow. This is something that we typically see with organisms that are called fastidious bacteria. Fastidious bacteria mean organisms that are tough to grow. They don't grow under general conditions. Selective and differential media. Now we're getting into some what we would call defined or synthetic medias. Selective media is going to contain ingredients that will actually specifically select for certain microorganisms to grow. A very common example that you will see throughout the course is a media called mannitol salt agar. This particular media has a high concentration of salt. Now, most microorganisms cannot tolerate high salt, but Staphylococci species can. So we use these mannitol salt agar plates to actually select for Staphylococci species. Now, take a minute and think about an area of your body where you have a very high salt content. And if you're sitting there thinking about this, you might come up with the skin. Your skin is very salty because of perspiration. Because of that high salt mantle that we find on the surface of the skin, believe it or not, the surface of your skin does not harbor a huge amount of different microorganisms. The primary organisms that we typically find on the surface of the skin are Staphylococci species. And the main reason for this is that they are able to actually tolerate that high salt concentration. So when we're selecting for, say, these particular organisms, we could take a swab and plate it onto a MSA plate, a mannitol salt agar plate, And the only things that really should grow up are staphylococci species because everything else would be inhibited. The other type of media is differential media. And what differential media will do is that it will actually differentiate between different bacteria, for example. So if you look at the example here, this is a general purpose media, a normal growth medium. And we have several different types of bacteria that have been inoculated onto this particular medium. And if you look at them, they all look white. And now if we use a differential media, okay, we can actually see that by using this particular medium, and this media contains different nutrients that can sort of accumulate different types of pigments depending on what the particular organism is eating, you start to see that you have these colonies growing up blue, this one growing up red, and then we have some white colonies. So you're able to differentiate based on how they grow. Okay, so again, selective media, this contains one or more agents that will inhibit the growth of certain microbes and encourage a select microbe to grow. This is very important in primary isolation of certain type of microorganisms from a mixed sample. Here are some examples of some selective medias. You'll see some of these throughout the course. Here's that one example that I used earlier, which is mannitol salt agar. Here's differential media. This allows for differentiation between microorganisms. And this is a media that's called chrome agar, and depending on the particular organism, the organism will grow up a different color. Here we can see pink, blue, a tan color, a lighter blue, pinkish, and then white, okay? And depending on, again, the organism, they'll grow up different colors. Okay, if you were to take all of these different bacteria and actually inoculate them on a complex media, they would all appear white. It's the media that is differentiating between these. And it might not actually be the bacteria that changes the color, it could actually be the media that changes the color. Here you can see, we have a normal tube here that's red. Here, the tube turned black, and in this case, the tube turned yellow, depending on the organism that's growing there, okay? So differential media is allowing multiple types of microorganisms to grow, but will display visible differences between the colonies. Another type of differential media is what we call blood agar. This is another type of media that you'll see throughout the course. Essentially what blood agar is it's composed of sheep's blood agar, okay, but it will allow various fastidious microorganisms to grow, and mainly because it has the blood serum. This is what these organisms need. And what you'll end up getting is these organisms will actually cause three types of hemolysis. Beta hemolysis, which is complete lysis of the red blood cells, alpha hemolysis, which is incomplete lysis of the red blood cells, and gamma hemolysis, which is no hemolysis. So if we look at this, you can see beta hemolysis here and here. And if you look, you can see the bacteria have pretty much lysed and broken down the red blood cells within the agar. You can pretty much see right through to the other side of this plate now, okay? Same thing here. If we look at alpha hemolysis, which is partial digestion, what you're seeing here is the bacteria is not completely lysing. Bacteria is not completely lysing the red blood cells here. And you're only seeing partial digestion. So you don't see that complete clearing. With gamma hemolysis, you just see growth, but you see absolutely no change in the blood agar whatsoever. Okay. Reducing media, this would be a media that will allow for anaerobic bacterial growth. Anaerobic means without oxygen. And then we also have carbohydrate fermentation media. This is testing for fermentation. Fermentation is a process of anaerobic respiration. It's where you can take a sugar, without the presence of oxygen and process that sugar. Unfortunately, because the electron transport chain is shut down and the TCA cycle shuts down, you end up going through a fermentative pathway where you will end up making an acid or alcohol from that sugar. As you go through fermentation and you produce the acid or alcohol, what will happen is the pH of the solution will start to decrease because you're making acid and alcohol. As the pH changes, the red dye that's located in this media starts to change colors. The more acidic the environment becomes, the lighter color this red dye becomes. And a lot of times, if you get full fermentation, the tube will turn yellow. So if you were trying to determine whether your bacteria could ferment a particular sugar, you could set up one of these fermentation assays, incubate it for 24 hours, come back the next day, and if you see that your tube turned yellow, you know that fermentation occurred because you produced some type of acid or alcohol. Otherwise, the pH would not have changed. That's the second example here. Other types of media, we have some media that are referred to as transport media. Transport media would be, let's say you go to the doctor's office, they take a swab, and they then package that swab to be sent to a lab. That swab will most likely enter into a, transport box or FedEx envelope, and then it will be transported to the facility, which could be a day or two later. And what the transport media's job is to essentially create a stable environment for the microorganism. You don't want it to grow, but you don't want it to die. You just want it to sort of stay in a static state. Okay. Okay. Another technique that's very commonly used is isolation technique. And what the isolation technique is taking a mixed sample of cells and being able to isolate them, say, on a petri dish to where you get individual colonies. Now, when you see a growth like this on a petri dish, this growth actually started from a single cell. And what happened is that single cell actually went under cell division and made many different clones of itself. And when you actually see a visible colony like this is not one cell, but rather this is billions and billions of cells that have grown from a single cell. Okay? So when you do something like the isolation technique, what happens is you're going to essentially start off with a very heavy number of bacteria, and you would say streak that out in a small portion of the plate here. Okay, and there is a lab that we're going to do next week that sort of goes over this technique. After you make that initial patch, which you can sort of see right here, and you can see it's very densely populated, you would then take your loop and sterilize it. And start with a sterile loop and then streak out another patch, just pulling a little bit from that initial patch. So what you're doing is you're dragging down a small subset of cells to a new area. And we can sort of see that right here. And you can see it's still pretty densely packed, but what you're starting to see is it's starting to thin out a little bit. Then you would actually do it a third time here, sterilize your loop. And now you would pull down from the second patch and create a third patch. Being careful not to touch back into that first patch, because if you touch in that first patch, you're going to drag down all the cells. And here you can see that it has really started to thin out. We're starting to see individual colonies here. And then you do this one last time, a fourth time, and you sort of streak out, down here from the third patch. Again, being careful not to touch back into the second or first patches. And you're dragging down a subset of that third patch. And again, you're pulling out even less cells, in which case now we can see individual colonies. Okay, we can see individual yellow, red, and white colonies present here. Okay. Now the requirements for isolation, this requires a small number of cells must be inoculated into a relatively large volume or expansive area of the media. You must have a relatively firm surface, a petri dish, an inoculating tool such as an inoculating loop. And like I said, this is going from a mixed culture to actually being able to isolate individual colonies. If you know that your initial culture is going to be very low in numbers, you could not do the streak plate isolation, but you could just do a spread plate where you just take a small sample and just spread it all over the plates. This would only work if you know that there's very few numbers of cells in your sample that you're plating. The idea here is to obtain a pure culture. Okay, a pure culture would be, if we go back here, seeing this red colony, there's no yellow, there's no white. Seeing this white colony, no red or yellow. Okay, these are pure cultures or pure colony isolation. Okay. Sometimes if you are not able to get isolated colonies, let's say it looks like this, and it's very tough to get an isolated colony. What you can do is try and hit the yellow area here and then repeat on a new plate, and you would kind of weed out any of the red or white that might actually still be there. Okay. One thing in microbiology is that when you are culturing, contamination is something you will have to deal with. As I mentioned, microorganisms can be present in the air. They can be present on any surfaces, so contamination could be a real thing. And even the best microbiologist will end up with contamination. So it's nothing that you should necessarily think that you did something wrong, but it's something that you want to try to minimize as much as possible. Okay. Inspection identification. We use various biochemical tests, and this is something that you'll see when we get to labs 7 and 8. That can be used to determine the fundamental chemical characteristics, such as what nutrients do they require, what products are given off during growth, are there the presence of any enzymes, and we can use these various biochemical tests to actually identify the bacteria. And like I said, we're going to focus on labs 7 and 8. We'll sort of focus on these biochemical tests. Other analytical and diagnostic tools. A lot of quick identification is either based on their DNA or RNA, and we can sequence those pieces of DNA and RNA pretty rapidly for quick identification. There's also antibody tests. You may have heard a lot about that in the news recently for COVID, as one of the tests that's being used to identify whether you are positive or negative. So there are other types of analytical and diagnostic tools that allow us to be able to identify these particular organisms. Generally speaking, though, if you're going to go down the culturing route, which is still the gold standard, it's going to take at least 24 to 48 hours. A lot of these, looking at the DNA or antibody tests, are much more rapid. Okay, looking at microbes by size, they are small, hence the term. Okay, and in order to see a lot of these things, we're going to have to use microscopes. Okay. Just to give you guys some perspective here, if you took anatomy, you probably talked about red blood cells, all right? Red blood cells are typically about 10 microns in diameter, which is 10 micrometers. Your typical bacteria is going to be about 10 times smaller than that, at about 1 micron. Okay. Now, bacteria, red blood cells, human cells, these are all cells that can be seen with a typical light microscope. And we're going to look at these different microscopes in a second. Viruses and some very small bacteria that are in the nanometer range, you're not going to be able to use a light microscope to see these. You're going to have to use an electron microscope. Okay. Now, most community colleges or two-year schools will not have one of these devices. They usually will just have light microscopes, as our institution just has these. So this is a typical part of a light microscope, and I'm sure everybody has used one of these before, okay, especially if you've taken biology or anatomy and physiology 1. If you've taken both, you've used the microscope in both of those courses. But as you'll see in lab exercise 2, which is due by the end of this week, it takes you through going through the parts of the microscope, how to use the microscope. So you guys will be working on sort of these areas this week with that particular lab exercise. Okay. Now there are 3 main properties of an effective microscope. One is magnification, making the object, magnifying it so that you can see it. Resolution, unfortunately, when you start to magnify, you will start to lose resolution. Okay, and you have to be able to account for that. Unfortunately, with the way that the properties of light are on the UV spectrum, we can only resolve between 2 points to a certain extent. So that's why with the light microscopy, we can only see down to about 200 nanometers. Because anything smaller than 200 nanometers, we can't distinguish between those points, just because of the properties of light. And that's why anything smaller than that, they no longer use a light microscope, but will use an electron microscope. Okay. So familiarize yourself with the different parts of the microscope. Again, you're going to be going through a lab exercise that sort of goes through this. Okay. Make sure you know the difference between ocular lenses, objective lenses. How do you figure out the overall magnification? Make sure you understand what resolution is and resolving power. Okay. And again, this should all be information that you should have touched on a little bit in the prior course, but again, it's also going to be touched on in that lab exercise that you'll be working on. Typically, and you guys unfortunately will not be doing this because this is an online course, but in an in-class lab, when you do get up to 100x or 200x, which is 1,000 or 2,000 total magnification, respectively, the light starts to scatter. And in order to refocus the light, we typically will use a drop of oil to help guide the light back up. So this is what I'm talking about here, is that if you use a lens where you're magnifying, 100 or 200x with an objective lens, the light just really starts to scatter all over the place. And what this will appear to look like when you're looking at it underneath the microscope is the image will be very cloudy or fuzzy. But by adding that drop of oil, we're able to refocus the light waves back up into the scope here, and you'll get a much clearer picture. Okay. You only need to use oil with anything above 100x, and that is if the lens is actually an oil immersion lens. They do now have microscopes now that do not require any oil. Okay, there are different types of light microscopy. We have bright field, dark field, phase contrast, and interference. The only one that we typically use in the lab for a course like this is bright field. Okay, but just take a minute to familiarize yourself with the definitions of dark field, phase contrast, and interference, and fluorescent microscopy, and where you might use those. This table here sort of goes through those different types, and it actually is nice because it gives you the same image. They're showing you the same image under the different types of microscopy. Keep in mind these are all light microscopes. They are all capped at 2000x, meaning you can only go above that. That's the total magnification you can go. And the main reason is because the resolution, with light caps out at 200 nanometers. Okay. Another type of microscopy is electron microscopy. And what electron microscopy has done is it has taken light out of the equation. Because we have that cap on the resolution here, we're now taking light out of the equation. By taking light out of the equation, we can actually now magnify up to, say, 100 million x, because the resolution is not 200 nanometers, but rather 0.5 nanometers. And essentially what they're using now is a beam of electrons instead of light, okay? And if the electrons get blocked, there's a filter on the other side, and if the electrons are being blocked from passing through because of the object, that would be reflected on the other side. Here's another electron microscopy image, and you can really see some nice fine details of the cells here. Okay, so again, just kind of go over and look over the different types of microscopy. Again, what we typically use in a microbiology lab is just going to be bright field light microscopy, okay? The only thing that you would not be able to see with bright field light microscopy is viruses, because they do fall below that threshold there of 2000x magnification. Okay, so now looking at specimens underneath the microscope, you need to prepare them, okay? And this is typically making the specimen slides. There are a few different things you can do. The first is what we call a wet mount. A wet mount is taking the organism, suspending it in water, placing that onto a microscope slide, and adding a cover slip and looking at it underneath the microscope. The advantages of using a wet mount: the cells are still alive, which means you can look at motility. You'll see the actual shape without any distortion. So those are the advantages. The disadvantages are that you can't actually stain for certain components for identification purposes. You can't actually stain to add contrast, which makes it easier to focus on. But unfortunately, when we move to the process of actually staining cells, we can't use a wet mount, and we actually have to do what's called a fixed slide. Fixed slide is preparing a bacterial smear where you would take a, say, drop of water, mix in some bacteria, let that dry, and then to fix it to the glass, you would pass it through a flame. By fixing it to the glass of the slide, when you then apply the stain and then rinse the stain, the cells will not go away. They will stick to the surface of the slide. Okay. Typically, like I said, we use heat for fixation. All right. A disadvantage of using a fixed slide is that the cells are dead. And because the cells are dead, you are not able to look at motility at all. Okay. Preparing different types of staining techniques. We have positive and negative stains. A positive stain is a stain that will stain the cells. And when you look at them underneath the microscope, you'll see different colored cells, depending on what type of stain you used. Negative stain is a stain in which you are staining everything but the cell. So you're essentially staining the entire background and not the cells. This sort of looks like if you were staring at it, stars in the night, where the stars would appear bright white because, or the cells would appear bright white because they're not stained, but the entire background is stained, say, in a dark black color. And here's an example of those two. Simple stains, these require a single dye. It's a pretty simple procedure. One of the lab exercises that you'll work with in this online course is you will look at simple staining. The main purpose of simple staining is just to add contrast to the cells so that it's easier to focus. You are not learning anything about the properties of the cell besides, like, is it gram positive, is it gram negative? That would not be something that you can determine from a simple stain. In order to obtain more information, you would do differential stains. And this would be a gram stain, acid-fast, spore stain, or capsule stain. These are different examples. And again, you're going to look at some of these as well through the online labs that we have set up for you. With the gram stain, this was developed about a century ago. It remains the universal diagnostic staining technique for bacteria, and essentially allows us to separate bacteria into two main groups, gram-positive bacteria and gram-negative bacteria. Essentially, with the gram stain procedure, which again you'll go through in one of the online labs, you'll go through each of the steps involved in the gram stain procedure, is you're essentially using two different dyes, a mordant and an alcohol wash to sort of end up with a final color here. And your two main options are either purple or pink. If the bacteria stains purple, it's gram positive. If the bacteria stains pink, it is gram negative. And we'll talk about why that is in the next chapter. Why are these cells staining purple and why are these cells staining pink? Okay, like I said, we'll get into that chapter with the next lecture, or we'll get into that topic with the next lecture. Acid-fast stain, this will differentiate acid-fast bacteria from non-acid-fast bacteria. Your acid-fast bacteria will stain pink and your non-acid-fast bacteria will stain purple. And one particular bacteria that it will stain with acid-fast stain is Mycobacterium tuberculosis. So if they suspect that you have TB, what they will do is they will take some culture, prepare a bacterial smear, do an acid-fast stain, and if they see all of these essentially pink rods, then it's positive for TB. Okay. Some bacteria are able to form spores, and we can stain for that. And again, we'll talk about what spores are in the next chapter. But spores are essentially hibernating cells, and they can live on surfaces for very long periods of time. A very common type of spore-forming bacteria, if some of you worked in the healthcare field, is Clostridium difficile, or more commonly referred to as C. diff. Whenever you have a patient that has C. diff, there's a lot of restrictions and precautions that are taken as far as contact and making sure that you are wearing appropriate PPE so that you don't essentially transfer that C. diff from one patient room to the next. But we can also look at spore development underneath the microscope with endospore stain by staining a particular stain that will actually, stain for the spores, and that's what you sort of see in this red color. These little red dots here are the spores, and the blue cells are just vegetative cells, which are non-spore forming cells. Other bacteria may have a capsule. The capsule is very important in protecting the bacteria from processes like phagocytosis. And we can actually stain to see if our bacteria of interest actually has a capsule. For this, we could do India ink, which is a typical negative stain. So this is where you're staining everything but the cells, okay, you can see the background gets stained with black here, and you can see that these cells have this white halo around them. That's because the capsule will not stain with the dye, so it's appearing white here, okay. A particular organism that typically forms a capsule is Streptococcus pneumoniae. Streptococcus pneumoniae causes respiratory infections which can lead to pneumonia. If they suspect you have pneumonia, that's probably the most likely cause for that pneumonia, either that or viral. What they will do is they will take some culture from the sputum from the lungs, and they can also do a quick capsule stain. If they see essentially capsulated cells, it's most likely Streptococcus pneumoniae. Okay, that is it for chapter 3, tools of the lab. Um, a lot of the techniques that we talked about today will also be parts of the labs that you guys will be doing over the next several weeks within this course. Okay, so you'll see a lot of these things sort of pop up again over the next few weeks, okay, in the lab exercises. All right. Have a good day.